associate professor at the departments of biomedical engineering and material science engineering in Carnegie Mellon. He received his bachelor's degree in materials engineering and Bachelor of Arts degree in chemistry from the Technion, and his master's degree in chemistry from Weizmann Institute, and his PhD in applied physics from Harvard University. He was a postdoctoral fellow at the Massachusetts Institute of Technology and Boston Children's Hospital in the labs of Bob Langer and Daniel Kohane. He received his 2012, he has many awards, the list is very long. He received the 2012 International Union of Pure and Applied Chemistry Young Chemist Award. 2014, he received Charles D. Kaufman Foundation Young Investigator Research Award. Next year, following year, he got NSF Career Award, ONR Young Investigator Award, George Talman Lab Research Award. He's also received Cellular and Molecular Bioengineering Young Investigator Award in 2018. With that, the floor is yours.
Thank you very much. Thank you very much for the invitation. I wish I could be in LA now, but we'll do it as we've been doing it in the last 18 months, I say, remotely. Today, I'll tell you my journey, or my team's journey, I should say, with bioelectronics and with nanocarbons. The work will be, the title is a bit different. It is more like, I have to admit, forming input-output interfaces with excitable cells and tissue using nanocarbons. So you may ask yourself, what the heck is I/O? So let me take one step backward and tell you about the matter we are interested to investigate. So on the biological end, you have these cells such as a neuron, as you can see over here, or a cardiomyocyte. These are soft and squishy. They communicate via fluxes of ions that go across ion channels that span across their membranes. These type of cells form networks, or, in the case of neurons, or syncytium in the case of cardiomyocytes, or we can arrange them in spheroids, organoids, or as in their native state, they are part of organs. On the other hand, when you talk about the materials that we are using, and others, I have to admit, we are using nanocarbons. Just few allotropes of these nanocarbons. You have the two-dimensional lattice of graphene, fullerenes, carbon nanotubes. These type of materials, one can control their electrical and optical properties. As you're aware of, they communicate via electrons or holes. One can attain them via different methods. We cook materials in my lab, and you'll see a bunch of the cooking we do. Usually we are doing things via gas phase synthesis. Other researchers use liquid-based synthesis or even laser scribing of carbon-based materials. One interesting aspect of these materials, since they have this enormous exposed surface to volume ratio, one can start anchoring molecules on their surface or modify their electrical and optical properties by anchoring molecules. So you can tailor their electrical properties. These are usually stiff materials, and one can form them in a soft and squishy manner. So there are a bunch of research groups that's what they do, making aerogels or making soft matter based on nanocarbons. That's not my research focus, but just as a side comment, one can actually form these soft and squishy interfaces. So what is I/O? For my end, or my interest, is actually our ability to sense electrical or chemical activity of cells. I wrote over here electroactive, but these are cells, not just electroactive cells. And you'll see close to the end of this talk why I just mentioned cells. It could be immune cells, for example. So sense the electrical chemical activity of cells, cellular assembly and tissues. That's the sense or the output. The input, which is the more on the actuation or modulation. So the ability to modulate cellular or tissue activity, either electrically or chemically. In the last several years, so I started my lab in 2014, January 10, 2014, we basically tried to push the limits of what we can do using materials. ultimate goal of the lab is actually to form this long-term investigation and modulation of cellular electrical activity. So we are en route toward that goal, to be honest. While taking these 7+ years of going through these 7+ years of journey, we came across unique materials that we learned how to cook in my lab, and we leveraged these materials to design several types of bioelectrical interfaces, such as electrodes that could be as small as an axon, so a couple of microns, give or take in size, or materials that have really unique optical properties, and we use them in order to stimulate without any tethers, just using light, or unique materials that we formulate in the lab that we can use for electrochemical catalysis or electrochemical sensing. And of course, some of the more on the bioengineering end is forming this 3-dimensional arrangement of sensors to basically detect the electrophysiology of organoids. Just before I delve deep into the matter, I have to admit that whatever I show you now, it's not these two hands that did the job. So I'm fortunate to have these people working with me. Actually, my group is kind of going through a grand change. So I have now many young students joining. I have currently, I think, 6 PhDs from MSc, 1 from BME, and a master's student from BME that is performing some research in my lab currently. So let's talk about forming input-output with engineered or 3-dimensional tissues. Actually, when we started working on it, we realized that there is a lack of capabilities when it comes to investigating 3-dimensional organoid spheroids. The reason there is a surge of interest in this type of cellular assemblies is basically stems from extensive literature showing that when you culture cells in 3 dimensions, they exhibit a structure and function that is closer to in vivo. I'm showing examples of neurons that are cultured in 3D. The same thing will be related to any other tissue, so cardiomyocytes, hepatocyte, etc. So there are hundreds or more than hundreds of papers just emphasizing this point. In this case, you can see 3D has more neurite extensions. 3D is more spherical rather than squished against the surface when it's a 2D culture. 3D will have a different cellular population and mortality. 3D cell culture will form more synapses across as compared to 2 dimensions. Okay, this is one aspect of the work, but if some of you may be aware of that, I think more than 10 years ago, as far as I remember, was a DARPA project that the sole reason of this DARPA project was to assemble 10 human, if I'm not wrong, 10 human systems on a chip, and then show that they can communicate. Some of the people on this call were part of some of the efforts or had their own independent efforts related to this idea. The thing about this part of track is the chip in most of the cases is just the mechanical support for the microfluidics or mechanical support for some of the electronics parts. So from my end, it is a huge step forward, but what we want to do is actually use the chip itself to be a functional and not just passive mechanical support. The big take-home message from this slide is that this line of research is not saturated. It's not over yet, to be honest. The big advantage of using this type of research technique, I call it now, is the ability to replace human tissues, or the ability to replace animal tissue and test directly compounds on human-based tissues. And the ability actually to investigate human tissue throughout extended time, let's say tissue development or disease progression, for example. So let me show you our idea. So this is Anna. She graduated a little bit more than a year from my lab, and she was the one that started this project. The whole idea behind her PhD work was just to get these three-dimensional assembly of sensors, active sensors, not just something that will be stuck to a surface. We call it organ on electronic chip. That's the reason it's organ on e-chip. It's not a typo. So the whole idea is just using simple material science to assemble sensors in 3 dimensions. What we are doing, we are generating a system that is like a spring-loaded system, basically. What you see over here is a array or microelectrode arrays, basically, on a sacrificial layer. And once you etch this sacrificial layer, because of pre-stressing metallic films, as I said, it's a spring-loaded system, the whole thing will pop out of the surface in a known manner. So this is a kind of pre-programmed self-rolled sensor array. So you can see that once you have this three-dimensional assembly of sensors, you can record, this is version 1.0, from the surface of this spheroid. In this case, it's cardiac spheroid. Whereas if you compare a planar MEA or microelectrode array, you will basically record just from the apex or from the bottom part of this tissue, and you don't have any knowledge of what's the status of the cell somewhere at the top, let's say. So this is the vision, and this is the reality. Basically, Anna mastered the fabrication of such devices. What you see over here, these are field effect transistors, so I will not elaborate more about that. But essentially, we can create these 17 sensors on a ribbon. You can see the dimensions are on the order of 200 microns by 500 microns. Once you etch this sacrificial layer, it self-rolls into a barrel-shaped sensor array. We can control the number of revolution by controlling the pre-stress or controlling the layer thickness, different layer thickness that we have over there. So, okay, this is attaining these sensors. So let me show you what we are doing with it. First attempt was to lock embryonic stem cells derived cardiomyocytes. This was done through collaboration with Professor Adam Feinberg in actually both of my departments. So this is a sausage-like spheroid, you can call it. The green labeling is the calcium indicator we're using for control basically recording. We recorded, you can see now the movie, the calcium transients as it beats. We recorded 12 sensors, and again, the calcium transient was recorded where exactly my laser pointer is. So a couple of insights about this type of interface. One can actually look at a single sensor now. So we have 12 sensors. Each of them is a different location on the surface of this spheroid, cardiac spheroid. And now we can analyze a single sensor. This is exactly what you see over here. You have 100 field potentials. So these are called extracellular field potentials. So that means that the sensor is outside of the cell membrane. So you're recording a capacitively coupled electrical signal that is generated. So basically each of these is a bitting event or an action potential event. So you take 100 bits and you average them. So this is the red trace. But something really unique about these field potentials, you can start extracting meaningful information, such as transients. So you can label sodium transient, calcium transient, and potassium transient. You'll see in a few more slides that this is kind of not the full orchestra of an action potential, and we have different techniques how to go around it. So this is a single sensor, but now we have 12 sensors dispersed in space. So one can actually look into propagation in 3 dimensions. So what you see over here is an isochronal map, but this is a 2-dimensional projection. You need to visualize it in 3D. It's kind of hard, but essentially you just fold it in a barrel shape. And this is exactly how it should look like. And from this one, we can actually extract the conduction velocity. So this type of approach is currently used in my lab to look into disease progression, for example. So we are currently working on various projects, trying to use similar approaches or different techniques to record in 3 dimensions and now start digging deep inside in the biology of certain diseases. So we are not limited in the type of tissues. Yeah? So one can use now brain tissues. So this is a mini brain or cortical spheroids. We basically aggregated or generated this spheroid by culturing these cells inside agarose microwells. And after 21 to 28 days in culture, we pulled one of these, again, locked it inside the self-rolled biosensor array, and started recording field potentials of spontaneous activity and actually evoked activity by applying a neuromodulator, in this case glutamate. So this is actually unique. What's unique about this approach is not just the recording itself. So the recording itself been done many years ago by other teams all across the globe, culturing cells directly on microelectrode arrays. In this case, we are actually trying to push the envelope toward organoid work. So this is a proof of concept, but ongoing or next step will be actually human-based brain organoids or developing brain organoids, I have to admit. The other aspect of this work that is posing it as unique is the fact that we took all this information or information technology that was developed in the last more than 20 years, I have to admit, and started looking into basically single unit isolation, spiking activity. So this is the firing rate as function of time, and basically raster plots to show the activity as function of time. This is the calcium recording. Actually, now the movie shows the fast blinking is due to the application of glutamate. It's around 4.5 minutes in this movie. After we published, or a day actually before we published our paper, John Rogers published a similar approach where he has assembloid so of cortical spheroids. So these are assembloids when you have two of them locked inside a three-dimensional platform as his team developed. What's really unique about his platform is the fact that it's not just recording as in passive electrode. He has different types of sensors over there or actuators such as temperature, light. So there's LEDs. So if you want to do optogenetics on these assembloids. So again, this is, I think, a beginning, I hope, of a surge of interest in assembling bioelectrical sensors in 3 dimensions toward basically tissue development, drug screening, disease progression on a dish in a 3-dimensional form rather than grow 2-dimensional cultures and use them. So let me just switch gears a little bit and tell you about some weird materials that the group is developing. I won't add much details about the materials. I try to tailor this talk be a little bit light on the material science, I have to admit. But one of the things that we heavily use in my lab is graphene. So this sp2 hybridized honeycomb assembly or crystal of carbon, which is essentially a single layer of graphite or basically a pencil, if you think about it. And so 2004, if I'll give a brief crash course on how to attain graphene, 2004, the Scotch tape method, I think they didn't get the Nobel Prize in 2010 for the Scotch tape method. They got it for the insane physics that came out of this work. And then there's a liquid-based method. So this is liquid exfoliation of graphite. So you get reduced graphene or graphene oxide, then you reduce it to get reduced graphene oxide. You have annealing of silicon carbide at 2000 degrees Celsius to get segregation of carbon at the surface, and they rearrange in a honeycomb lattice, so graphene basically, or chemical vapor deposition using transition metal catalyst. Most of it, this one was introduced early 2000s, so around 2007 or 9. My lab is using this type of method extensively as well. There were attempts to assemble graphene in 3 dimensions in the last several years, such as crumpled graphene oxide on silica particles, or growth of graphene on germanium nanowires, or basically a growth of graphene on nickel foam. I argue that no matter how you look at these, still the graphene is exhibiting a 2-dimensional topology. And I'll show you in a bit the materials we generate so you'll understand. The reason I transition toward this type of topic, because the next several topics I will cover are related to how to form input, so how to send signals into cells and tissue. So we came across a unique assembly of graphene that we call it fuzzy graphene 4 years ago. This is actually outcome of these two students, so Raghav and Sahil, and then Daniel joined, where we showed that we can actually generate something that looks like that. I don't know if you can see what I'm showing, but this is literally a pie brush, but think about this divided by 10,000 times. So these are nano pie brushes made out of a core that is a silicon nanowire, but again, the core could be any material that can sustain the growth temperature, which is several hundred degrees Celsius. And the bristles of the pie brush are basically single to fuller graphene flakes. So you can imagine that this structure has enormous surface area. And something really unique that we came across while working on this material, it has really unique electrochemical / catalysis properties and optical properties. So just by growing this material, this actually opened up so many research tracks in the lab. I'll present a couple of them in this talk. The first one I want to talk about is actually related to the fact that we do want to send information into cells and tissue, basically actuate cells or stimulate cells. So I'll give you a brief overview how it's done or how things are done, I have to admit, in the last many years. So you all probably fully familiar with optogenetics, basically expressing of channel rhodopsin in cell membranes. These are algae-based ion channels that are light sensitive. You shine laser on them, you get the channels to open up basically, and then you can elicit an action potential. One big hurdle of this technique is the fact that you need to genetically modify the organism in order to get these ion channels to be expressed. And of course, this will potentially hamper translation in the future, or clinical translation, in other words. The other technique that was developed somewhere, I think 2010, give or take, is actually using IR lasers and focusing IR lasers on tissue volume, and pulses of IR lasers can actually change the cell capacitance. So this is optocapacitive effect, and you can elicit an action potential. So this all or none behavior of cells. In this case, it was first demonstrated on neurons, and then even, if I'm not wrong, cardiac tissue as well. This type of approach is limited by the resolution and actually the needed energy to get an effect. There's a very limited threshold for approval of using this type of light tissue interfaces. And you can see the amount, I have it in other diagram as far as I remember. It shows that millijoules range. This is how much it's using. Other techniques that are based on nanomaterials, such as gold nanoparticles, silicon-based particles were developed. These are outstanding approaches from a very close friend that he actually trained my lab on some of the techniques. This is Bo Jatian from U Chicago. Some of the limitations of nanoparticles or even polymer mediated is related to limited wavelength. So these materials, it's not a broadband absorber, for example. Limited absorption, so that means that you'll need to crank up the energy or the intensity of your light source to get an effect. And limited material stability, so polymers or even silicon particles can decompose after some time once injected. Some applications, you want to have a transient effect. So you want to have the particles in and then they'll disappear after a few weeks. So that may be an advantage, not just disadvantage. Let me show you how we are doing it. So we are generating a library of optically tunable materials. We call it hybrid material because we are using a silicon as the template, and then we grow a shell of graphene, as I mentioned before. And this graphene shell, we can control the density, the size, and actually the chemistry of these graphene flakes as well. We can also control the nature of this core. So I will show only one demonstration of either heavily doped or intrinsic silicon nanowire, but currently we're working on optoelectric devices such as PN junctions. So the idea behind it, you take one of these, let's say you have one of these brushes, you stick it on a cell, and you shine pulses of laser, and you hope to see activation or modulation of electrical activity. These are the library of materials we generated. You can see that we can literally tailor the density of the flakes and the size. You can see that the scale bar is changing. And this is just by controlling the time of the synthesis. Again, it's a plasma-enhanced chemical vapor deposition process we are doing. You can control the density and size of flakes by controlling the temperature of the process as well. So we take one of these wires and actually, I'll skip, let me go back. Basically, something really unique about these materials that are kind of opposite of graphene. So graphene should be 97.4, if I'm not wrong, or 97.6 transmittance. In our case, it's 97% absorbance. This is this structure. A mesh that is composed of these wires will have close to 97% absorbance. So it's basically black material, not super black as a highly oriented carbon nanotube, vertically oriented carbon nanotube, but I say it is a broadband and a very efficient absorber. So if you take now if you shine pulses of laser, let's say 1 millisecond pulse of laser, and we are using glass capillary technique to measure the temperature, one can measure temperature rise of up to 7 degrees Kelvin. When you do the same measurement on a bare silicon, you get barely 0.35, if I'm not wrong, 0.35 degrees. And you need to crank up the laser power to get this effect. The spot size is on the order of 20 microns, by the way. If we basically plot our materials as in the energy that is needed to get a temperature versus temperature rise, you can see that the materials we are using, such as NT3DFG, NT3DFD is this one, is a lot more efficient than the rest of the materials out there, such as silica coated gold nanorods, gold nanoparticles, silicon nanowires, and gold decorated silicon nanowires. There are a couple more materials over here that I will show one slide. This is just hot off the press. Literally was released last week. This is using Maxine, which is another flake van der Waals or flake material that we started using a few months ago, or I should say a bit more than a year ago. So here is how we are interfacing these wires. So you take this fuzzy wire, you alter the surface of the wire with a self-assembled peptide to render it to positively charged, and then it will stick to the cell membrane. I said that the wires are black. The reason you see them white over here, because we reverse the contrast, so it will be you'll be able to see it in this confocal image, laser scanning confocal image. Just the cross-sectional of these laser confocal image, laser scanning confocal image. You can see that the wires are just attached to the cell membrane outside. If you take the same cell culture and dry the cells, load them in the SCM, you can see the same thing. But I did not mention these are dorsal root ganglion cells, basically. So the wires are stuck to the outer membrane of the cells. So here is the experiment, just to show you that one can encode information in neurons. We patch the cells, so this is a whole cell patch, and here is a fuzzy wire attached to the DRG membrane. We had pulses of laser with fixed time, so pulse width of 1.2 millisecond, and we changed the pulse power. So in other words, we gradually increase the energy per pulse. And what you can see over here is really nice transition toward the threshold. This is basically the membrane potential from which you have this all or none event of action potential. Once we cross this threshold, we get an action potential. And the whole idea behind it, or the mechanism, is the optocapacitive effect. So there's a whole literature explaining this effect. Basically, the membrane capacitance is changing proportionally to the temperature change, essentially, with time. So the larger the temperature change, the larger the membrane capacitance change. And there is, as I said, capacitively coupled current. This is the part of the optocapacitive effect. And this current is either due to charges that are being generated, so we don't have this part in our equation, or changes in the membrane capacitance. And this is exactly what I just mentioned. The larger this value will be, the DC to DT, the larger IC will be. And this is exactly what we were able to show by performing a control experiment, where you hold the cell membrane at fairly negative values. So I'm talking about minus 150 to between minus 150 to minus 120 millivolts. So the cell is seriously hyperpolarized. And then you shine this laser pulse, and of course it's in a voltage clamp, so you can see the current that is being generated while we shine this laser pulse. The values that we get for our materials, and again, in this case, I show you only the NT3DFG, are the lowest reported energy per pulse of 1 millisecond pulse, if I compare it to the whole various literature out there. So this is this is an outstanding hybrid nanomaterial to be used, to be honest. I'll show you the Maxine, how does it look in a bit. But Maxine is more efficient. I have to admit, the film is as efficient as NT3DFG, and flakes, which is kind of insane. It's a 2 nanometer thick, less than 2 nanometer thick flake, let's say 10 to 20 microns in size. And it's as efficient as other nanomaterials out there, let's say silica particle or porous silicon particles, for example. One can drive a neuron multiple times. So this is a ground truth measurement where we use the capillary to stimulate the cell, and then 200 milliseconds later, we apply a laser pulse and get the photothermal stimulation of the neuron. We can drive the neuron in different frequencies. So we can do a stimulation every second, every 200, we did half a second, 200 milliseconds. So I just show 1, 10, and 20 hertz, but we did in between as well. In 20 hertz, it kind of falls apart. So you can see these tiny action potentials, just because this particular cell is limited due to a refractory phase of the ion channels, sodium ion channels, most likely. So one can alter the electrophysiology of a network. So you have a network of DRG over here, and we stimulate number 1, and you can see how the calcium signal propagates across the cells. And now you can basically plot the fluorescence as function of time, and you can see the propagation from one to the other. This is not a synapse-based propagation. This is more of a chemical propagation. That's the reason the time scale is fairly long, and it is supported in literature as well. Something really unique about this approach is that we are not limited to two-dimensional. We are not limited to a single cell. So one can sparkle it on organoids or spheroids. One can mix it in with a certain threshold below the toxicity. As we all know, we need to test it, and we've done it. So in this case, you can see NT3DFG mixed in cortical spheroids, and you can see that the particles are dispersed all over the spheroid. This one is literally sprinkled, if you can imagine, like basically like a cupcake. We sprinkled it on top, but we have other data showing that we can mix it in as well. And we used the same spheroid and selected one cell within the spheroid and basically used a laser pulse and showed that we can stimulate the whole network while doing so. The Maxine is really unique, I think, in a way, and I'm not going to present all the data we acquired with Maxine. It's just accepted now in a ACS Nano. And the big advantage of Maxine is actually the large scale production that one can get. So I'll be honest, the NT3DFG, these spy brushes, we can do, if we'll push it, milligrams a day. The Maxine, you can do kilograms a day if you have a large scale production. So if you think about incorporating such materials in additive manufacturing, etc., this is a huge plus. Another big plus of this material is the absorption peak. which is around 800 nanometer. And this is exactly kind of a textbook a wavelength, or the first transparency window for the body, where you have lower absorption of hemoglobin and water, basically. So we showed that we can generate films of MXene, and you can see over here the wrinkles in the film. The film is 90 nanometers in thickness, and it's kind of a large scale film. So it's a few centimeters. You can make it a few centimeters across, or we can actually do flakes. This is what I mentioned before. So 2 nanometers in thickness, and they are on the order of few microns to, let's say, 10 microns, give or take. It depends on the synthesis conditions. This was done with collaboration with Yuri Gigotsi in Drexel University. So you can see in this case, the MXene is attached to the cell membrane, and same idea, we shine pulses of laser and we can get cell actuation. So something that is not sending in information, but more utilizing optical properties of materials is related to, again, another aspect of bioelectronics and the need for new types of large-scale patch clamp. I'll explain as I go. What you'll see over here is actually a collaboration between my team and De Angelis in Italy, in IIT in Italy, where we basically utilized his capabilities to optoporate using materials and my team's capability to generate these unique hybrid nanomaterials, or in this case, the fuzzy graphene. Actually, what we tried to do initially was just to show that one can use this, the unique optical of this material to porate a cell, but then we realized that the process is so efficient that you can actually stimulate or optoporate multiple cells in a few seconds. So let me take one step backward. This is an optical patch clamp system. That's what it is. So if any of you is doing patch clamping, you know that probably takes you more than a few minutes to patch a single cell. In this case, 9 cells in a few seconds. So we are using ultra-fast laser to raster on the electrodes, and we are using such conditions that we generate cavitation. So it's a different process as compared to the photothermal stimulation. This cavitation generates tiny holes in the cell membrane and give us access to the cytosol, and then we have intracellular-like action potential. So this is how the multi-electrode arrays or fuzzy electrodes look. Again, these are 5 microns by 5 micron electrodes that we are generating. I'll skip through this. So this is extracellular field potentials, and then you shine pulses of laser and you get these intracellular-like action potentials. What's unique about this approach, now one can screen for drugs and have the whole orchestra of ion channels. So as I said, the extracellular field potentials are highly useful, but they're limited. They don't show all the currents that are part of the orchestra of the action potential. Unlike extracellular field potentials, these intracellular-like action potentials give you all the orchestra of ion channels. And this is one example. We tested a bunch of, I think 2 or 3 compounds, and this is still ongoing work across the labs. So in the last few minutes, I want to talk about I think the convergence, because what I showed you, it kind of seems like eclectic. He showed us like bioelectrical platforms, some materials for photostimulation, some materials for large surface area electrodes. So here is a challenge that basically literally confluence of everything that the lab is doing. This is a collaboration across multiple institutes. So UPIT is leading us. I'm leading TA1 and 2, and TA3 is a researcher in UPIT is leading. I'm working with Rice, Northwestern, and University of Pittsburgh, basically, and of course, researchers within Carnegie Mellon University. The sole purpose of this program is actually to hack biology in regeneration of muscle. So I'm not going through all the biology, but essentially, if you have a large volumetric muscle loss, you will end up with large scar and very little muscle growth, and even techniques that are based on biomaterials that are kind of far more advanced won't generate a large repaired muscle, let's say, or functional muscle. The aim behind our approach is to cut the time by 50% for muscle regeneration and get 75 muscle that is functional. The way we are going to do it is basically by sending in chemical, electrical cues and sensing. So you see, this is the confluence, because my team knows how to generate these electrical stimulators and sensors, essentially. The teams in RISE are Omid Weisse, Jacob Robinson, and Ashok, and I always blank on his surname. He will forgive me. In Northwestern, this is Jonathan Rivney. He's the guy that is working with me. We collaborate extensively, and you'll see in a bit on what. So the sense, one of the things we try to sense is actually the immune status of the muscle after this big defect that we generate. And I'm not talking about a skin or a shallow defect. I'm talking about 10 centimeters by 4 centimeters by 3 to 4 centimeters deep muscle loss in a canine model. And our aim is to actually gauge the state of the macrophages within this wound. And we design different types of electrodes. I'm just showing fuzzy graphene-based electrodes, and show that we can actually generate these highly selective, highly sensitive microelectrode arrays that can sense nitric oxide. And we are actually translating these currently to in vivo. So as I speak, we are performing in vivo experiments toward that goal of sensing the state of the wound. So while you know the state of the wound, I want to modulate it, or I want to modulate, let's say, reinnervation of this large muscle loss. The way to do it is by actuation. So some of the materials I showed you are used toward that goal. So high surface area electrodes that we generate and hybrid nanomaterials that the lab is generating are currently being used across the labs to stimulate muscle and nerve tissue. And then, of course, we are following it over a long course of time to see whether it does affect the reinnervation. So this is just to summarize the materials we are generating. Basically, the metrics that usually we look at are the impedance of these materials or the charge injection capacitance, and then the capacity retention. Our materials actually improve current capabilities of kind of more standard material that is used in bioelectronics, which is P.PSS. And actually, even fuzzy graphene by itself is highly stable and actually has increased capacitance because of oxidation of the surface or pseudo capacitance. So this is the current state of affairs. I can show you that the same materials that we generate actually can be used to record the state of the muscle. So I can send in information using electrical stimulators, but now I can generate this array. And I was about to show it and I forgot. So all this I show is assembled. I don't know if you can see it. It's a 10 or less than 10 micron thick polymeric layer with all these sensors and actuators on it. This is the in vivo model we are using now. In this case, you can see, I'm sorry about the gore images, but this is the large muscle defect in a canine model, again, done with Steve Badilak's lab. And you have an array of electrophysiological sensors. And we show that our sensors can pick up evoked response. So there is a nerve stimulator, sciatic nerve on the sciatic nerve, and we measure evoked response, and we can actually start construct something that is like a textbook diagram of a recruitment curve of muscle activity. The aim behind this study is to follow it over a long course of time to show whether any electrical cues we provide will change the recruitment curve. Is it changing per site on the muscle as well? This is ongoing work. This is actually the only unpublished piece of information. We are trying to wrap up the last in vivo experiments when it comes to this type of experiments. So this is exciting. It's 7 years of confluence, literally, in one massive effort toward muscle regeneration. So I'll summarize this talk. I just showed you a bunch of technologies, materials we are using, so I will not go through all the details all over again. I just need to thank a bunch of people. This is actually taken yesterday in my CVD room, the chemical vapor deposition system. We call ourselves hybrid nanomaterials chefs. So that's the reason we have these weird, funny hats. If you're interested in cooking hybrid nanomaterials, we have openings over here. We are seriously looking for eager and enthusiastic people to work with us. If you have interest in collaborating with us, same idea. So I'll take questions now, and thank you for the opportunity, and thank you for inviting me.
Excellent talk, Professor Cohen-Karni. Appreciate it. So I'm waiting for questions. You've shown many technologies, including optogenetics, nanowires. One thing I didn't understand, the PIN diodes or PIN devices you place into the cells. You excite them with a laser. You can also excite them with electrical signals like wiring.
Yeah, this will be tetherless. This will be just a diode. So this is work. This part of the talk is work in progress. Actually, Boje already showed it in his own work. We are trying to see whether coating this will make them more efficient, coating this with graphene. So essentially what will happen, if you shine light on this standalone structure, you'll have charge separation. You'll have actually electrons in the shell, and you'll have holes in the core. And this will actually will depolarize the membrane by mirror charges on the membrane side. You'll have the same idea of transient current forming.
I see. Thank you. Any application on neurodegenerative diseases?
Oh, this is a great question. So I think this one definitely should be applied, and yeah, I'm biased, should be applied in such research track. We should be able to generate disease on a dish in 3D. So the answer is yes. Stimulators, one can investigate whether they reverse effects or change course of disease as well.
Thank you. The flexible electrodes that you showed in the beginning, they nicely wrapped around organoids. But
yes.
It's mainly the surface. Have you thought about somehow integrating into the organoids?
We are currently, so this is a great question. Two comments about it. First comment is one potentially can use surface recordings to infer the state of the inside of the tissue. This is called the source localization. So if you know what's the structure that you're using. So this is part of a project we just started. We're trying to use surface recording in 3 dimensions to infer the state of the inner part of the tissue. Second part of the answer is yes, we are thinking about new ways to assemble sensors. Other researchers, such as, let's say, Zhao Yu in Harvard University, and Zhenan Bao in Stanford, they try to assemble sensors in organoids as well. We try to take a different angle to see whether we can have highly ordered arrays within tissues. You have work by Charles Lieber, Tal Dvir, Boje, as well as early paper from his postdoc, looking into basically assembling sensors in more like a folded tissue. So this is more like a patch-like approach. We are not working on patches. We try to work on organoids.
Thank you. There's another question. What about chemical stability of the coating on these nanowires?
The graphene is fairly stable. If this is what whoever asked it meant.
No, I think they're saying that if you have a nanowire, the PIN, other things, you may put a coating around it to make it compatible with cells. And is that stable?
This didn't test it. So what we did is just in this case, if you refer to this idea I mentioned over here, we used the technique developed by Dr. Wanunu in Northeastern, and we self-assembled the KF peptide just to render the surface of these wires through pi-pi stacking to be positively charged. We never tested the stability. The graphene itself is highly stable. So we have data out there showing how chemically it is stable and electrochemically stable as well, which is kind of surprising, to be honest, because the notion was like, you have so many edges, it will probably degrade so fast. And we have a paper out there showing that you can electrochemically cycle it for millions of cycles, and the chemical properties do not alter that much. That means that the flakes do not degrade over like 7 million cycles of fast scan CV, for example.
Are there plans to make biodegradable wires and devices or implantable devices? Looks like most of your efforts are like in vitro.
Oh, that's good. That's good.
I know Chris Bettinger is doing a lot of biodegradable things, but
yeah, but you have John Rogers. He's the leader when it comes to transient electronics. I say he had a bunch of papers showing stimulators. I think his most recent work is electrical stimulators that just disappear over time. So it is possible. I haven't explored it in my own team yet, but it's possible.
Okay. For the 3D self-rolling bioelectronics, how do you extract the signal? Is it with wires or wireless?
It's wires. We are using, so I didn't put all these details. We are using Intantech amplifiers. So let's say I have this over here. You probably can't see it, I assume, but this is one typical chip that we use. And this chip will have connection, a printed circuit board that will be connected to Intan technology amplifier. And it's the one we are using is usually 32 channels, but you can buy systems that are like 10, 24 channels from them. I'm not doing like commercials from them. They're one of the most one of the cheaper options out there, I have to admit.
Okay, thank you. Is N-Xylene biocompatible, or does it have any safety concerns, especially for long-term use on implantable application?
Which material?
N-Xylene, M-X-E-N-E.
MXene.
Yeah,
we ran the typical live dead and actually even phototoxicity assays. It is fairly safe to use, to be honest. And we are not the only one that reported it. So other labs looked into its use. The titanium carbide MXene has been used extensively in phototherapy, as in laser tumor ablation. So unlike this research track, we are not trying to ablate anything. We are trying to stimulate. So we use different energy regime.
Thank you. For drug screening applications, how do you plan on integrating nanowires into microfluidics?
The drug screening, you can actually, we don't have it currently in the lab, but part of the agenda is if I'm switching to this one or this idea, you can, because it's top-down microfabrication, you can definitely have a flow chamber over there, a microfluidic that will flow your compounds. So this is something, so all these assays were done by pipetting in and out chemicals. This is actually not pipetting. We have a flow chamber, but a large chamber as this chamber. Let me open this dish so you will see exactly what I mean. It's like a 3D printed, oh, come on. When you need it to open, it does not open. So this is a 3D printed, I don't know if you can see it,
yeah,
but this is a 3D printed frost room, like a chamber, and we have line in and out, literally tubing, and we use gravity to flow, gravity and vacuum, gravity to flow and vacuum to put it out. So that's a large scale, and we checked it within a few seconds. You'll remove the entire volume. It depends on your flow rate and the level of and the amount of volume you have within the chamber. So not necessarily you need a micro channel. What you need is actually for long-term measurements, you need something that will be in aseptic conditions. So this is something that we are working on.
Thank you. Heterostructures are a good candidate for photodynamic therapy. What is your idea about using the potential of this method in combination to photothermal, e.g. for manipulation on the cell membrane probe?
So this is a great question. You guys ask good questions. And I'm not giving excuses now. So this is something that we literally started now. Like the first experiments in materials characterization are done as I speak. The way I see it, it will have similar effect as the photothermal effect. So you basically have pulses of laser and you will generate these currents within the cell that will elicit an action potential. There are other approaches for such structures to generate small molecules. So I'm maybe going sideways now, but you can do photocatalysis and you can actually generate small molecules that could alter cellular behavior. For example, you can generate peroxide, hydrogen peroxide in small levels, not just to form any large ROS response in the cell. So this is something on the agenda as well.
Thank you. Does the ruffled surface of the graphene contribute to the potential fouling effect when applying for sensing applications?
So the graphene is not exposed. So what we have, I don't have this data, and actually my student showed it to me yesterday. We have data about fouling of sensors after several days in serum media. The graphene itself is not exposed because it's coated with various polymeric layers that serve as selectivity layers. So we are generating, the graphene is just the collector, the current collector. And we have some, so if you look, I have the diagram over here, I think, yeah. So we have the graphene is the high surface area electrode, and we have some sensing layer. This is a catalyst we are using. And then we are using some selectivity layers. I put amino naphthol as one of them, but we have other layers we are testing as well. So the graphene is fairly protected from the solution. So we are actually looking into fouling of the selectivity layers. Our application is limited for a few days. So we are not doing long-term implantation for now for the wound healing project.
Thank you. Does your nanowires and other nanomaterials have good adhesion with cells? Do they attach properly?
I think they are, because I'm biased, but they are. You can see them; they attach. We do like essentially wash experiments, and they still stick to the surface after we wash. Let's say, if this is an indication of attachment, we never try to tug on a wire to see if it will pull. Yeah, but once we disperse them, they attach to the cell.
Yes. Well, that's all the questions I have. Thank you for your time and the great presentation, Tzahi.
Thank you very much. Stay safe, I say.
You too.